Hello everyone. This is the fourth part of our plant hormone and carrot tissue culture experiment, and this is day 14 after introducing the carrot tissue onto the culture medium. Our first experiment where we were producing callus. These are all the petri dishes with the introduced tissue, and they look good. I don't see any contamination. And if we look up close, we see that they're continuing to swell and change shape. And we also see what could be the beginnings of friable callus tissue. Now, friable, it just means that it isn't attached. It comes apart easily and can be pulled off of the rest of the tissue. So it's easily detached. And that's what callus is. Callus is undifferentiated tissue. Undifferentiated plant cells. So they are not committed to any sort of developmental pathway, and they can form, and they're pluripotent and totipotent, and can develop into whatever sort of organ that is needed. In a whole plant, the only equivalent of callus would be probably meristem, and of course in a whole plant there's not a lot of meristem. It occurs in just a few layers of cells in very specific places of the plant, yet it's incredibly important because it is responsible for the growth of the entire plant itself. Here is an up-close picture where we can see the friable cells. And right here, and you see these little blobs right here. We can't say for sure. It's a little early to say that this is callus, but just from experience, I can say it sure looks like it, and it looks like it's an early stage of callus production. To remind ourselves of our progression, we have our day 0 where we had chunks of blocks of carrot tissue. Day 7, they're starting to swell. And day 14, they're continuing to change shape. And we think, or it's possible, that we're beginning to see some callus development. In the second experiment, where we were testing 16 different auxin and cytokinin ratios, and it was kind of a test, or it was experimental, or I'll say, ends up all were contaminated. So that experiment is no more. And what we see at day 14, week 2, is all were contaminated. We have mold growing in a lot of them, and in others we have more mold, and in others we have even more mold. And our ants were back. But all of the tubes were contaminated, so that experiment is just gone. It's just a failure. And of course, anytime an experiment fails, for whatever reason, you want to think back to what was done and ask yourself why, and see if you can think of a good answer, or see if you can pinpoint something that was the reason why it didn't turn out the way you expected. And in an experiment like this, the reason for the failure or the unexpected result had nothing to do with a dependent or independent variable that you were testing or using as a control. It really is just about technique. So we need to review the technique and see if we can come up with reasonable answers for why we had contamination. So first off, you go back and you question everything you did. Was a sterile technique during media preparation? And this is where having really good documentation of what you did comes in handy. This particular experiment, of course, you want to write everything down, but I have pictures of everything too, so we can go back and review. So first off, was it sterile technique when I was preparing the media? And what I come up with for this is that the Petri dishes are fine. So, and the media was all prepared at the same time. So since the Petri dishes are not contaminated, it probably had nothing to do with media preparation. So the next step was where tissue was introduced into the culture media, and this could have been the point where contamination was introduced. But once again, this was all done at the same time. So since the Petri dishes are okay, it probably was not when the tissue was introduced to the media. So what about the type of culture vessel? Because so far, all I can think is that the Petri dishes are okay, but the test tubes are not. So what's different about those? Well, the tubes, of course, were not parafilmed, but the Petri dishes were. So could that be it? I mean, it's a possibility. I mean, I can't say for sure, but the parafilm did prevent ants from crawling into the Petri dishes like they did into the culture tubes, and maybe ants crawled into all the tubes, and they just weren't there when I went in to take the pictures. And of course, ants would introduce any manner of contamination. Perhaps it's air movement. So there's no air movement in and out of the Petri dishes since they're sealed. The test tubes were not sealed. However, these are designed to prevent air movement. And so the cap goes on, and air would have to go all the way up around and down like this to get into the tube, which is unlikely, but still, you never know. I mean, the ants got in, so maybe there's some kind of air movement. Also, I've used these tubes and caps a lot of times, and I've never had the contamination problem, the sort of wholesale universal contamination problem that I saw this time. So I'm still questioning what in the world was it. We can also question the incubation conditions. In this case, I left everything out on a bench. That's what the protocol suggested, was just to leave everything out on a bench. And if we were doing this in a class setting, the students would have just left it out on a bench out of the way in the back of the lab. So I was trying to recreate that. Perhaps it was that. Usually, if I'm doing a tissue culture experiment, I have an incubator where the air is still, right? There's no fan or a significant amount of airflow in the incubator. It's just a place that holds a constant temperature. So at the end, I don't know. I have no idea why all of them were contaminated. I can only come up with ideas and hypotheses. And if I was to do this again, I think what I would do is just not use the test tubes and just stick with Petri dishes. So our next, in about a week, we'll have day 21 data, and so log back in for that.